All right, so I'll talk today about reproducible NGS data analysis with Nextflow and nfcore. And you might wonder, why do we put so much emphasis at QBiC for reproducible data analysis? So we all would probably agree that reproducibility is very important in science. It's in fact probably the foundation for science. And in the last decades, there have been quite some articles that have scrutinized how reproducible published experiments actually are. In a pretty famous paper by now in 2016 by Baker in Nature, they have interviewed 1,500 scientists and asked the question if they ever failed to reproduce an experiment. And across scientists in diverse fields in science, the answer was always the same, that more than half of the scientists actually had some problems reproducing their own published results or someone else's published results at some point. And the vast majority of those scientists also agreed that there is a reproducibility crisis in science at the moment. In another paper in 2009 by Dionysius et al. In Nature Genetics, they also analyzed how easy it was to reproduce microarray results that were already published. And they found out that more than half of the experiments from those papers could not be reproduced, or the results from this paper, sorry, could not be reproduced. And they also analyzed what were the reasons for this lack of reproducibility. And in more than half of the cases, the reason was that the sequencing data was not made publicly available. But other common reasons for reproducibility issues were that the software to run the analysis was not available, the methods were unclear, or simply a different result was obtained. So when going into the field of NGS data analysis and bioinformatics data analysis in general, there can be several reasons or several factors that can contribute that the whole analysis procedure is not reproducible. If we take, for example, the typical metagenomics data analysis that Daniel has already introduced in the previous talk, we would see that the typical analysis procedure includes several steps. For example, adapter trimming, post-read removal, assembly, binning, and taxonomic classification. And for each of the individual steps, several tools can be used for data analysis. So one first factor, obvious factor that will affect the reproducibility of the results is to know exactly which tool and version of that tool was used for each of the analysis steps. But that's not the only thing by far. Each of the individual tools can accept different parameters. So it's also important to know what parameters were used to run the analysis. Also, if the tools depend on other software for their functioning, the dependency versions will also be important. And it has lately been also seen that the runtime environment, so the system libraries, or like the place where this analysis, like the infrastructure where this analysis is run, is also can affect also the final results that are obtained. So there are several measures that can be taken to ensure reproducibility in NGS data analysis. Starting from a naive point of view, one could think that, for example, a step-by-step documentation would already improve reproducibility. But this is typically not the case, because having a very detailed step-by-step documentation of the analysis is tedious, and it's often incomplete and insufficient to reproduce the analysis. One extra measure would be to share the code that was used to perform the whole data analysis procedure. But it has been seen in many cases that code is not sufficient. Sometimes code is difficult to understand or difficult to install. So code accompanied by sufficient documentation on how to run the pipeline and what actually the data analysis procedure is doing is important. And it's also very important that the code is easy to install by other researchers, so other people can also reproduce analysis results. But finally, also introduced before that the runtime environment is also important to obtain the exact same results when running an analysis. So even this would not be sufficient, but there should also be some additional measure that ensures that the analysis is always performed on the same environment. And to facilitate achieving this gold standard of reproducibility in bioinformatics data analysis, there have been lately the concept of reproducibility data analysis, reproducible data analysis on workflows. Reproducible data analysis pipelines or workflows. Both words are used interchangeably here. So what are analysis pipelines or workflows? Basically, they are nothing else than the code that comprises all of the steps that are necessary for the whole analysis procedure. And analysis pipelines are usually organized in processes where each of the processes calls the necessary tool for a particular analysis step. To facilitate writing this kind of code, there have been appearing several specialized programming languages for writing these data analysis pipelines. You might have heard about the common workflow language, such as that is also abbreviated CWL, or SnakeMake, or Nextflow. Those are all specialized programming languages that facilitate writing data analysis pipelines for bioinformatics. And to ensure that those pipelines are reproducible and can be run, and the same results can be obtained in different infrastructure, all of the tools and dependencies that are needed to run the analysis pipeline need to be packaged together with it. And to achieve reproducibility, the state of the art at the moment is using containers. Containers are similar concepts as virtual machines and make it easy to ship all of the necessary tool dependencies to run the pipelines, fixing the versions and fixing also the runtime environment so that the analysis can run in different infrastructure. So each of these specialized programming languages have their advantages and disadvantages, and we at QBiC have committed to writing all of our data analysis pipelines using the Nextflow workflow language. This has several reasons. One of the most important ones is that it is committed to ensure reproducibility. Nextflow supports multiple container engines that I've mentioned before are pretty important for reproducibility. And we at QBiC use both the Docker or Singularity container engines, depending on where we run the analysis pipeline. An additional feature that is important for us from Nextflow is that it makes it easy to run the analysis pipelines in different infrastructure. So without having to change the pipeline code, we can run the same analysis in local computers or high performance computing clusters or the cloud by just changing the executor from Nextflow. An additional feature of Nextflow is that it contains continuous checkpoints for the intermediate files and procedures. So it allows pipeline resuming whenever one of the process or step of the pipeline is failing. One can resume the pipeline in an intermediate state. And it also provides a feature that is called implicit parallelism, which means with the same pipeline code, we can process a different amount of samples, and all of these samples will be processed in a parallel fashion to allow using as many resources as possible at the same time. We are definitely not alone in the world that are using Nextflow. There's a huge community that uses Nextflow to run data analysis pipelines. And here in this map is summarized some of the institutions that are currently using Nextflow. And together with these institutions, we are part of a project called nfcore, which stands for Nextflow Core Pipelines. And the purpose of this project is to collect a curated set of Nextflow pipelines for different bioinformatic analysis purposes that are community curated. So nfcore uses Nextflow for, like, it's a collection of Nextflow pipelines, but it also provides additional guidelines that ensure even more that the data analysis pipelines are state-of-the-art and reproducible. One of the important features is that each of the nfcore pipelines should have stable releases. This tags a specific version of the pipeline. So when using a stable release, one can always reproduce the same, one can go back to the old code even when new versions of this pipeline have already been introduced. Another feature of the nfcore pipelines is that they should contain extensive documentation on how to run the pipeline and also on what kind of tools the pipeline includes, which makes it easier for other people to reproduce the analysis. Additionally, to ensure that no bugs are introduced for each new feature or new piece of code that is introduced to a pipeline, there are some tests that are running that are called continuous integration. And this is basically a small data set that runs through the whole pipeline. And only when those tests run properly, then this new piece of code or this new feature is introduced into, it's merged into the pipeline code. As I mentioned before, containers are an important part for reproducibility of data analysis. So all of the nfcore pipelines come with all the packaged software that is necessary to run the analysis, and several containers are supported also as part of nfcore pipelines, which makes the analysis portable. We can run them in our own laptops. We can run them on high performance computing clusters in case that high memory is needed, or in different cloud infrastructures. And towards the cloud direction, we have recently introduced a feature which is for each new pipeline release. The pipeline will also be tested on real size data sets on the Amazon cloud infrastructure to ensure that the pipeline also runs properly on full size data and that the analysis is also cloud ready, so it can be performed also in the cloud. So those are a bit more like the technical properties of the nfcore project, but a very important component of the nfcore project is also the community. So at the moment, there are close to 300 contributors to the nfcore pipeline, which means that many people use these pipelines and also contribute to these pipelines. So this ensures that they are easy to use for other people, and also if some problems or bugs appears, that those will be reported and fixed in a quick manner. Also, the principle of nfcore is not that everybody shares their own pipeline, but that there is basically just one pipeline per analysis type. So for example, one pipeline for implicit analysis, one pipeline for metagenomics analysis, and these ensures collaboration of different members of the community that are interested in the same analysis type, ensures that the pipelines will be best practice, so many people can use it, and ensures that other scientists that are also interested in the same type of analysis will actually review the code to ensure a high quality of the analysis pipelines and on the developed code. So we at QBiC are contributing actively to several pipelines that are part of the nfcore community, especially in the metagenomics and microbiology research. We are actively contributing to the nfcore map pipeline, which was recently published in bioRxiv, the nfcore implicit pipeline, which Daniel already introduced before, but also back as a bacterial assembly pipeline. In other research areas, we're also actively contributing in pipelines in topics of immunoinformatics for epitope prediction, HLA typing, MHC peptide identification, TCR and PCR analysis. And in the area of genomics and transcriptomics, we also actively contribute to a variant calling pipeline or single-cell RNA-seq data analysis pipeline. So let me show you a bit in more detail how does this look like, how many different pipelines the nfcore community contain. So we have a nice website, the nfcore website, and here there's a list of all of the pipelines that are currently available there. So currently there are 32 different pipelines that have stable releases, and through the nfcore website one can also have a closer look at any of them. So when clicking on one of the pipelines, you're redirected to the pipeline-specific documentation, pipeline-specific page, and this page contains general documentation about the pipeline, so a schematic on what does the pipeline actually do, quick start on how to run the pipeline command, and yeah, basically general information about the pipeline. The usage documentation explains in more detail how the pipeline will be run, what are the input files that are needed to run the pipeline, and how this can be provided, and some further documentation also on how to run the pipeline, basically in general. There is also a specific parameter doc page, in which it is explained all of the parameters that can be used to run the pipeline and what do they do, what do they alter. And finally, there's also an output documentation page in which it is explained, once you've run the pipeline, inside the results folder, exactly what are the different subfolders that can be observed there, and what each of the files there represents. So I have also mentioned before that for each pipeline release, we run the results on full-size data sets. And here on the nfcore website, this is also used to showcase example results that one can expect when running that analysis pipeline. Here, for example, when running the map pipeline, there's an example of the interactive taxonomy graph that it's obtained when running the Corona tool. So what are the prerequisites actually to run any nfcore or any Nextflow pipeline? One, just Nextflow requires Java, so it should be installed in the system. Then it also requires the Nextflow installation itself. And if containers are used, which are recommended for reproducibility, also the container engine should be installed, in that case, Singularity, Docker, or the container system that is preferred. It also needs to be noted that natively supported at the moment are only macOS or like Unix systems, so macOS or Linux. So once these prerequisites are installed, to run a Nextflow pipeline, it can be done from the command line, and basically the command would be Nextflow run, the name of the pipeline, the version that one wants to use, and different configuration can be also provided. Here it's the installation of the specific pipeline is not needed, because when this pipeline is the code for the pipeline is openly available on GitHub, Nextflow will directly pull the pipeline code and the containers that are needed from GitHub. So it doesn't require any installation for each of the analysis types. Then when running this on the command line, one can see first what are the default options for the pipeline. One has to say that for all of the nfcore pipelines, there are sensible defaults, but some specific knowledge about all of the tools that the pipeline is running is also needed in case somebody wants to adapt the analysis pipeline specifically to the task at hand. So this is an example on how to run the small size test for the nfcore map pipeline. It would take a while to run, but basically here Nextflow also logs what processes are running at the moment, if for how many samples they are running, and if the process has completed or not. The same developers of Nextflow have also developed Nextflow Tower, which is a monitoring system that can be used for any kind of Nextflow pipeline. And we are using it also as part of QBiC to monitor all the pipelines that are running at the moment in our cluster system. There you can see if the pipeline ran successfully, if there was any error, in which step the error occurred, and the error message. So it helps us basically keeping track of all of the analysis that are running simultaneously at QBiC, and if we have any problems, to be able to identify it quickly. So I hope I could summarize here why do we have this emphasis on reproducibility at QBiC, why reproducibility is important for us, and why it should be taken into account for any kind of data analysis in bioinformatics. For us, it's important also to use Nextflow for our data analysis procedures to ensure that we can scale our analysis types to large data sets, and we can parallelize it across our available infrastructure. And we are also happy to be part of the nfcore community, because we benefit from this large community for pipeline development and peer review. And with this, I'd like to end my talk and thank everybody at QBiC, and especially the research development and data science team, which is mostly the ones developing this Nextflow pipelines and nfcore pipelines. And I would also like to say a big thanks to the whole nfcore community. In case you are interested in more details about nfcore, here you have a QR code that you can scan, and we'll show you, if you like, how to join nfcore and a bit more information about this international project. So thanks very much for your attention, and I'm happy to take any questions now.